Muhammad Umar Farooq Biological Video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem in the comment below. Here we will discuss about the bacteriology. So the bacteriology is the branch of biology which study about the bacteria. So let's talk about the little bit overview of the bacteria. And that's first of all I want to say that the bacteria is the prokaryotes. Prokaryotes mean the bacteria which that is without nucleus. On the other hand, eukaryotes, which that is the true nucleus are present like a human. So that is the monera is the it is kingdom monera which that is classified. So prokaryotes without nucleus including all bacteria. So important to know that the bacteria contain a two type of DNA. First of all, that is the DNA. First of all, I want to say that the eubacteria and archaeobacteria, archaeobacteria, it's known. So the eubacteria means true. The bacteria will have with cell wall. Murine are called eubacteria. On the other hand, it is contain murine cell wall. While archaeobacteria is the ancient. It means the ancient. It means the old time it's developed, and that is without murine cell wall. So their difference, it is also known as the false bacteria because it is not true. While that is the bacteria here we will discuss that is the without murine cell wall. So the bacterial structure is contain a two type of DNA. First is the plasmid, which that is a smaller circular DNA is here, and the big circular DNA, which that in the form of nucleoid is known as the DNA. It is without nucleus, and that is present in the cytoplasm, but the nuclear origin is present is look like that. It is present in the center of the bacterial cell. So at the cell wall, and cell wall is made up of peptidoglycan. So the peptidoglycan we will next diagram we will discuss. So first of all, we should understand about the introduction of the bacteria and what is the bacteria. So here is a Louis Pasteur's contribution during the 1867. So that is the first of all he had described about the bacteria, what is bacteria. So the fermentation is the process which that is provided by the Louis Pasteur. So the in 1880 in 1867, Louis Pasteur, which that is demonstrated the role of bacteria in fermentation and decay. So that's important character is the decay of the material, organic material. So the Pasteur evolved many basic techniques of bacteriology. He described that bacteria cause disease. So, and the second one we will discuss is the vaccine formation. So the anthrax, fall, cholera, rabies, that is the all infection caused by the bacteria is produced by the Louis Pasteur. So that's famous due to. So the pasteurization is the another step which that is introduced by the Louis Pasteur. It also a great development of pasteurization. So the pasteurization is the process in which milk or other liquid are heated at certain temperature, for example 60 degrees Celsius, in a particular time, for example 30 minutes, to kill the bacteria. So these is the important things which that is bacteria maintain in normal temperature, and their environment of the bacteria is the normal. So the according to the temperature, every bacteria are present in the specific region on the basis of temperature, while the pH is also. So and salt resistance bacteria. Halophile. So the halophile means the loving to the salt, while the acidophile is the acidic medium. So the acidobacteria. and Bazobacteria, as well as the halobacteria, which that is on the basis of acid toward the moving of acid. So important to know that here is the Robert Koch and German theory 1876, which provide the postulate about disease causing agent. Number one is the a specific pathogen or organism uh, present in specific disease, so that is the specific disease. On the other hand, second is the pure culture can infect to the animal when inoculated into animal. Pure culture will can infect to another susceptible animal, while the third one is the recovery is possible of the organism, so which is infected in pure culture as experimental basis, so it can be curable, while the viral disease cannot be curable, like a AIDS, HIV. So here is the shape of bacteria, which that is first we will discuss, and we will discuss one by one the overview and the information about the bacteria according to the bacteriology. So here is the cocci, that is the oval shaped bacteria. So the oval shape is bead like structure are present, while bacilli, which that is the bacillus structure, rod shaped bacteria. So that means the rod shaped bacteria is looking like this, while the spiral means the spiral shaped curved shaped bacteria. So that's here is the spiral look like this, while the variety of shape, a different variety of shape of bacteria is known as the pleomorphic. So here we will discuss also the spherical cocci, which that is the spherical. It is also known as the cocci. So the different type of cocci is in the form of spherical, which is it can be single or attached with the to make a colony. While here is the two bead which interconnect to each other to make a diplococcus or diplococci. While cocci is the coccus, it is known as single, while the cocci it is multiple of coccus. Here is the streptococci. Streptococcus mean the thread-like structure which is made up of cocci. So coccus. So here is the tetrad and sarcina is also present. Sarcina means the four containing bead is known as the tetrad, while eight is known as the sarcina. And on the other hand, staphylococcus is the multiple beads which that make a colony, while their bacilli or bacillus is look like this, diplobacillus and triplobacillus, while the streptobacillus is made up of thread. So remember, blue-green algae is the major example of it, which that is the blue-green algae is a look like that. While here we will discuss about also occurrence where the bacteria are present. So today where the bacteria are present in everywhere, by the variety of bacteria is can be different. So the variety is depend, while here is the water which contain salmonella typhosa, which that infect the digestive tract, vibrio coma shaped, which that is looking like this, and this vibrio are coma shape. While in the food, meat, egg, which contain Asexual Proteus. So the Proteus mean the protein related to the protein. That is mean about this. While in air, that is the Sarcina species and Clostridium, which that is look like this, Clostridium and Sarcina species, which that are present, and their structure is look like this. While on the other hand, in the milk, it is also the bacteria are present in the milk, which that is a streptococcus and lactobacillus, while the E. coli is also. So the lactobacillus is produce the lactic acid during the if we will not boil the milk. So the lactobacillus will be produce the lactic acid to to spoil the milk and uh, change the quality of milk. so convert into the yogurt is also used by the lactobacillus or E. coli. So the different bacteria produce the different type of yogurt on the basis of company which that is provide the lactobacillus type and E. coli type of yogurt. While here is the human, which also contain a bacteria, but its infected bacteria as a parasite are on the other hand um, helpful microbiota in our gut. So the salmonella and mycobacterium it are also present in the mycobacterium tuberculosis are present in the lungs which infect the lungs while the salmonella is present in our digestive system which damage the digestive system to cause diarrhea, while here's the diplococcus and clostridium tetani which that produce the tetanus, while the pneumonia is cause the pneumonia in respiratory pneumonia or any other. So E. coli is also. On the other hand, plant which that is also contain a different type of bacteria, and soil is also contain a different type of bacteria. But here also we will discuss the just overview about the name of the bacteria which infect the which that are present in the soil used as a mutualistic behavior with the plant and soil for the nitrogen fixing bacteria. So the fix the nitrogen by the fixing nitrogen fixing bacteria, while the plant contain a xanthomonas, rhizobium, and uh, uh, arvenia. That is different type of plant containing bacteria. But here we will not discuss about the infection of the plant, but on the soil which that's contain a different type of bacteria which that is used for the nitrogen fixation for ammonification, and that is nitrogen cycle we will discuss in the next video. So here is the nitrosomonas, nitrobacters, rhizobium, and azotobacter which that is mutually interconnect to each other to form a ammonium ion production from the nitrogen oxide and nitrogen dioxide and simple nitrogen to fix to give the plant to make a protein. So amino acid contain an ammonium ion. So the nitrogen containing group is known as the protein amino acid. While here is the general structure of bacteria we will discuss with the briefly, and also life cycle as well as the indication of gram positive and gram negative bacteria. So let's talk about the here I am drawing the structure of the bacteria is just simple. We are not going to understand the mesosome and their organelle as well as cytoplasm. Just we will discuss about the structure as well as the reproduction while the lifestyle of the bacteria. So we will not go into the deep, and next video I hope I will make a new video about the transcription and translation as well as the antibiotic effect on the bacteria cell wall and rupturing. So let's talk about the here is the structure. Here is contain a disease causing finger-like projection is known as a fimbriae, while conjugation or sexual reproduction use the pili. So the pili is made up of pillin protein, while the fimbriae is another, and that is flagella which that is contain a flagellin. So the flagella is the long thread-like structure which used for the movement, while the pili is also used for the attachment in the substratum, right? And here is the DNA. Circular DNA is also known as a nucleoid, or we can say that is the one chromosome containing bacteria. So here is the plasmid. Plasmid is the extracellular DNA which that is also present in our mitochondria in the form of circular is look like that plasmid, while that is look like that plasmid circular many circular DNA, while this is the plasmid which that is used for the transformation from one bacteria to another. It is also used as a vector which that is used for the production of insulin in a biotechnological field. So here we will also discuss about the cell wall. So the cell wall is made up of peptidoglycan in gram positive. Here is the gram positive cell wall, while here is the gram negative cell wall. So the gram positive cell wall is the thicker as compared to the gram negative wall, which that is the thinner, regard to the peptidoglycan, not regard to the cytoplasm, because cell membrane. So the cell membrane is the double in the gram negative, while gram positive cell wall is made up. Gram positive cell wall is a thicker but a single layer. So here you can say that the blue color lining shows that the teichoic acid and lipoteichoic acid, which that is invaginate in it to make a solidity or something, while the peptidoglycan is the red color, shows that the peptidoglycan chains, which that is interconnect to each other. Here is the important to know that the peptidoglycan layer is a smaller and thinner as compared to the gram positive cell, while upper part is made up of the bronze lipoprotein. So this bronze lipoprotein is attached with the plasma membrane in the outside of the phos- bi phospholipid bilayer, which is also attached by the porine. So the porine is the protein channel containing pore containing protein, while the red color shows that the upper line is known as the lipopolysaccharide. Lipopoly mean lipo mean lipid, and poly mean many, saccharon mean sugar. So the many chain of the sugar are present, so on the upper side, while in the gram positive cell, which that is not lack of this lipopolysaccharide. So the difference according to the structure is here, while we will also discuss about the gram positive and gram negative on the basis of color production by the experiment, while here is the hydrophilic head is present in the phospholipid bilayer and hydrophobic tail, which that is interconnected to each other. Tail is connect to each other, and the hydrophilic head will be show inside and outside, which that is used for the preservation of electrolyte, etc. While here is the capsule, which that avoid the phagocytosis of the macrophage, and this avoiding is the mechanism which that infect our immune system uh, will be no response, but the antibody will be produced to engulf easily. But next we will discuss about the bacteria as an antigen to provide the antibody by the immune cells, while here we will discuss about the asexual reproduction first. Next we will discuss about the sexual reproduction in bacteria. So asexual reproduction is known as a binary fission. Here is the parent cell of bacteria, which that's convert into the daughter cell. So the parent cell and daughter cell is contain equal amount and equal genetic material, which that is the phenotype is the same, and genotype is also. While here we will discuss about the conjugation, which that is also known as the sexual reproduction in bacteria. So here's the another bacteria which that is attached by the sex pili. So these sex pili is made up of pillin, and that facilitate the movement of plasmid. So suppose this plasmid is one cell, it's provide the plasmid is known as the cell F positive, while which that receive the plasmid is known as the F negative. So this mean here is the plasmid, which that is known as the F plasmid, which that provide their DNA by this mechanism. How it's possible? We will discuss. So here is the transferosome, which that provide, and here is the DNA polymerase, which produce the DNA, which that is same look like a F positive bacteria which contain it, and transferosome, which that is bind uh, with the replicated DNA, which that is a complementary DNA template, which that's go into the plasmid and insert to the plasmid, and either insert into the plasmid of the F negative cell or F negative cell is lack of the plasmid, so the plasmid develop into the F negative cell. So this plasmid is same look like the F positive. So that means, what is the meaning of this? That is that genetically recombinant bacteria will be formed for the using of environmental effect to survive. So that different changes of the environment will be allowed to provide the new genetic recombinant bacteria. So this is the natural recombinant bacteria by the conjugation bridge. So here the conjugation bridge will be formed. I hope you make sense. So it is sexual reproduction which that provide the DNA by the F positive to F negative, which that is the lack of the plasmid, or the plasmid which contain a DNA it's our own but insert the DNA from the F positive. So that's the same things. So you should understand that is the lack of plasmid are present in the recipient DNA. So it's a recipient bacteria. So these bacteria is two type, a donor and recipient. Donor is donator and recipient is the recipient receiver. So the donation and receive the plasmid is by this mechanism. While here we will also discuss about the identification of gram-positive and gram-negative bacteria, how to identify. So here the first of all, the crystal violet is provide, that's the mode and the first, and that's the all purple color will be show in the slide. While when we will also provide the iodine solution after the washing out, and this iodine solution will be provide the show the all purple gram positive and gram negative both bacteria show in the purple color, while gram positive purple and gram negative is the colorless after the using alcohol and after the washing to use alcohol, that show the gram positive in the form of purple, while gram negative is the colorless after the using of the alcohol, while safranin is used after that mechanism that's provide the color. Gram positive show the gram positive show the purple color, while the gram negative show the red pink color or red or pink color. So that we can identify the bacterial bacteria forms either either it is gram positive or gram negative bacteria. So next here we will also discuss about the sexual reproduction and which that is used by the third party. It is known as the bacterial transduction. So the bacterial transduction is by the bacteriophage. So this bacteriophage is attached by the bacterial cell, and this attachment is wind up by the fourth step. So the fourth step, first is the landing and the second is the adsorption. Not absorption, adsorption. Adsorption means the bacteria will remain outside, not absorb. So the third one is the penetration. Penetration means the penetration of the tail by the using of enzyme, and that's releasing mechanism which that release the viral DNA into the cytoplasm of the bacteria. So what will be happen? So this viral DNA will be inserted by the DNA ligase enzyme used to it is also known as the endonuclease, which that is cutting of the DNA and attachment of the foreign DNA with a viral DNA with the bacteria DNA to produce this shape, and this is known as the recombinant bacteria. Which that is why recombinant bacteria, because bacteriophage is insert their DNA in it, while this will be growth as bacterial cell wall and produce the head and tail region as well as the DNA which will be splitting, so that the lysis will be occur. It is also due to the endonuclease enzyme used to it to produce this function, and after the rupturing that's produced a lot of bacteriophage that will be infect again. So this is known as the Lytic cycle, because the lysis will be occur. So the lytic is related refer to the lysis. So the virulent phase if bacteria infected by virus. So this is the virulent phase. And if we will talk about the non-virulent phase, that is also known as the lysogenic cycle. Lysogenic means the basically lyso means the inserted DNA, and genic means the generation. So the more generation will be produced by these recombinant bacteria, lysogenic bacteria. So this will be not lysogenic because the environment is unfavorable. So these bacteria will be divided while the favorable environment will be preferred to the Lytic cycle. So this will be reproduction, asexual reproduction, to produce more and more recombinant bacteria, and ultimately when environment will be favorable, that will be lysis. So the next is here we will discuss about the bacterial growth. So here I am drawing the graph which that shows the bacterial growth. So the vertical column, vertical line shows that the log number of bacteria. It means the increasing of the bacteria is at power by 4, 5, 6, 7, 8 and 9, 10. While is the horizontal is shows that the hours. So the 5 hour, 10 hours and 5 hour, 15 hours, 20 and 25, which what will be happen. Here the 5 to 1 to 5 hour is used for the lag phase. It is used for the preparation, while the log phase is used for the rapid growth. So that cell division will be occur, asexual reproduction. So the colony will be grow. This colony after the 5 hour later cover the 10 hours, that will be stationary phase. Stationary phase is between the 5 hour and during the 15 hour, that will be stationary. And after this stationary phase, the bacteria will maintain their death rate and reproduction rate is equal. So the cell division is equal. While after the 20 hours and 25, that will be death and decline phase. Rapidly bacteria will be die. So this is the bacterial growth curve shows that. While here we will discuss about the also nutritional lifestyle. For example, the bacterial lifestyle according to the nutrition. So this is the lifestyle which is parasite. Parasite bacteria get foods by host infection. So that's infection causing agent is known as the parasite. So that is known as the heterotroph. While if we will talk about the autotrophs, which that is first we will discuss about the Litho Autotrophs. Litho Autotrophs means that is lithosphere will be withering, and after the withering the consume the calcium ion etc silicon, which that is used as a energy, and that's conversion of the Litho Autotrophs convert this energy, and in the byproduct that's withering will the process by the bacteria. So that is known as the biological withering. While photoautotrophs is that autotrophs which produce glucose by the using of light. So these bacteria will be produce glucose with own cell by the using of light and water. So and carbon dioxide basically. So chemoautotroph is used chemical to produce energy, while the sulfur fixing bacteria use sulfur for the fixation of instead of carbon dioxide, so instead of water. So here is next video we will also discuss about the explanation of sulfur fixing bacteria, but here the symbiosis will be occur due to the bacteria is known as the microbiome in our gut as well as another animal. Also skin is also contain bacteria which that can be harmful or non-harmful. Mutualistic behavior, commensalism, which that is neither harmful nor benefit, but bacteria is benefit itself, while parasitism is the harmful effect which that bacteria grow and get benefit, but the host is harmful for it. So here is the long chain fatty acid to short chain fatty acid production, for example propionate and acetone etc, which that is the helpful and mutualistic behavior in our gut is known as the microbiota. So thanks for watching. Please subscribe the channel and give the suggestion for new video, and coming soon. I will make a new video. So thanks. I hope you make sense about this. Bye.